many different people. And I think from here, I will give a hand to, when I show you how we analyze it, I will give a hand to Tommi to show him actually that he will show you what we do with the data afterwards. Thanks.
Thanks, Carolina, for a nice presentation. Yeah, before I start, I would like to just mention that we will have some time for questions also in the end, or discussion. So just keep in mind, if you have any questions at this stage, you can already write some in the text chat box, but you can also raise your hand in the end. Yeah, I will share my screen. Let's see. There we go. So I will present some statistical approaches and workflows for analyzing metabolomics data. I will start by telling a little bit about pre-processing the raw data, how that goes into a more simple format. Then I'll talk about modeling approaches, starting with metabolic modeling, which is a more complex approach, going into enrichment analysis of pathways. What does that mean? How does it work? And then finally, I will tell about lipids and the lipidome and how those are special among the metabolites. So first, about processing, as Carolina also showed in some slides, we get from mass spectrometry this kind of overview of data. So we have different masses, compounds of different masses coming through the chromatography at different times, and each of these gray dots in this map are potentially a unique compound. We don't know at this stage, but that's possible. And then we need to crunch these into something meaningful. So yeah, if we look at one single point here, that's basically a scan in time of different masses. And when we integrate this data over time, we get peaks, which represent compounds. So that's the signal. And then we need algorithms to crunch this into an Excel table, or this kind of simple format where we have participants and compounds and levels, concentrations of compounds. So this is a rather complex area of science itself. We need algorithms to get this signal into numbers, and we need databases to identify the compounds. What is each of these dots that we don't know to start with? And when we get to this point, we can then start working on the data and ask questions. Are there some metabolites that change in the disease? And so in data analysis, I will start with the metabolic modeling, where the idea really is just to have a map of the phenomenon. So we have the metabolic network that you could consider as a map like this, where we have roads that lead somewhere and cross, intersect each other. And in this map, we have reactions between molecules, the crossroads, and in the crossroads we have substrates and catalysts or enzymes in biology usually, and products, what comes out of the reaction. And then there's a rate of this reaction. So every junction in this network is a sort of a reaction. Something goes in and something goes out. This is also roughly how metabolism works. And then, so that's just the framework, but when we measure data, we get concentrations of this network, or the traffic in the network. So what is exactly happening within the roads? And that's basically omics. We measure compounds and we try to find out where in the network there is something happening, something unwanted happening in disease or in treatment, how the network is responding. Yeah, so one example study of this kind is this paper where the authors looked at alcoholic fatty liver disease by integrating multiple omics, gene expression, proteomics, metabolomics into the metabolic network of liver cells. And by doing that, they identified what is changed in the disease in the network, where the balance is gone wrong, and they could find that certain amino acids are basically shut down, or there's a change in the synthesis of these. So blue arrows happening here. So we have less traffic than what we would expect in a healthy cell. And yeah, as a follow-up, this study was taken forward. Like the results first were based on data, but then when the authors knew what is wrong in the network, they did a simulation study that how can we fix the network? So let's fix the roads in a way that there's better flow through the network. And they took some key metabolites of the network, handful of those, and simulated if we bring more of that into the cells, could that fix fatty liver disease? So could the change in the metabolism by bringing in these compounds, more of that, lead to better balance of fat in the liver? And they simulated this, so adding different amounts of these compounds, and eventually found a balance where each of these reactions is in a normal state again. So that was a sort of theoretical way of trying to solve this question. And then once this optimal recipe was figured out, they did a small clinical trial, feeding in this mix of metabolites and see can that actually change the liver phenotype. And yeah, this is not fully published yet, but there is some first results about that in arXiv. But in a very small study of participants, feeding those right metabolites for the participants every day, they could see some changes in the liver health measures. So yeah, some pilot example of how to translate multi-omics results and complicated metabolic networks into something that could work as a treatment. But yeah, I will switch gears then to enrichment analysis, which is basically a simplification of metabolic modeling. So this is, like I said, rather complicated approach, and often we are not really measuring all the compounds in the network. So there are just so many metabolites, like Carolina mentioned, and those cannot really easily be measured, and we need many different methods to do that. And then again, secondly, this is only a snapshot in time. So this might be a network of 4 p.m. traffic, but we all know that it's very different from 9 p.m. when it's a different flow. This is the same thing in disease. Different stages look different, and also in when you take a pill, half an hour later, 2 hours later, or 1 week later, they're different phenotypes. So these kind of variation bring some obstacles to these modeling approaches. So we can simplify it by enrichment analysis, where we just really look at one neighborhood at a time. So we don't need to know the reactions exactly, and like what goes in and what goes out, but we know that these metabolites are acting together in some way. And if we measure enough of those, we can figure out, is this group of metabolites somehow changed? And this is an example of then pathway category. We have a neighborhood in the map where we see more red than what we expect on the whole map. So we can infer that there's something going on here. There's an enrichment of traffic. That's essentially what we are doing in omics. Also, when we do enrichment analysis, we look at the different parts of the network and find out, is there something unexpected happening? And how is it done in practice? Well, there are like different databases where we can find information about metabolites and other biomolecules. This is an example that also was mentioned in the previous talks, HMDP, Human Metabolome Database, which basically covers most or really many metabolites and biomolecules. And it shows information like this. We have molecule called valine, which is an amino acid, and then we have different information. And we have also information about the pathways that it participates in, about diseases where it's changed, and about like treatments or drugs which activate or deactivate this compound. So for example, here in this page, you can see different pathways that it's involved in. You can click through, see them visually, and understand what is the context. And but when we do it in data analysis, of course, we do it in a more automated way. We don't do it manually going through everything, but we bring this data into the algorithm and then crunch the numbers, find out are there many molecules in one pathway that are changed. So brief example, here we had also a study on liver fibrosis in rats, where they measured different metabolites after the start of the disease. So we see some changes in the metabolites over time. And when we bring in this pathway information and do the enrichment analysis, we get a result like this, where each square is a rectangle, sorry, circle is a one pathway, and y-axis shows the significance of that pathway. So we have some valine amino acid pathways changed, as is shown here in these individual metabolites. And then we have a lot of pathways that are not really changed. They are just here around zero. So some individual metabolites might be changing in those two, but not really overall. And the most significant is sphingolipid metabolism, which are lipids that are changing. Here appears to go reduced in the disease. And lipids, they are actually some particular type of compounds. This is a sphingolipid, where it has a head group and a fatty acid chains. So there are many lipids of the same kind with the same head, but they are of different size and different saturation. So there are different lengths and different numbers of these unsaturated bonds. And this information you can find on lipid maps, which is one database on lipids. So here I'm listing all the sphingomyelins. It's about 100 different ones that have very similar name, but just different numbers. So they are different length and saturation. This one is SM38-1, so it has 38 length and 1 double bond. Yeah, so lipids, it's not so meaningful to study them in the same way as metabolites. They're basically all part of the same pathways, so it's not really very informative. But we can look at them in this way, for example, where we put them, categorize them by size and unsaturation. So these same properties here are on x-axis and y-axis, and we show basically the change in disease in color, and each rectangle is one lipid species. So we see, for example, SM38-1 here in 38 and 1. About here, and that one is blue, so it is reduced in fibrosis at Kleiner stage 4. And we see basically this kind of pattern where larger sphingomyelins are reduced and some smaller ones are increased in the amount in liver disease. This result is from Galaxy, actually, and by doing this more systematically, we can look at many lipid classes like sphingomyelins, as I showed, phosphatidylcholines at different stages of disease. So going from more healthy liver onwards to fibrotic liver. And yeah, this data we have integrated with other omics, gene expression, gene regulation, and also lately like a Mendelian randomization study to validate some of these findings. And these type of analysis you can do with Lipidomer, which is actually an R package that I have created. You can install it with this command and find some examples on the web page. So with that, I'll just wrap up. So we need processing of the data to start with, and we can use metabolic models to explain the metabolic networks. And when we measure omics, we can actually understand what is going on in the network, what is changing. Enrichment analysis is basically a simplification of this approach, where we just look at groups of metabolites together and try to see, do they change coherently or not? And then finally, lipids are a bit particular type of metabolites, and those we can look for patterns in size and saturation within each class. And these parameters might be something that is relevant for disease. So thanks for this session. I will just